Good morning. Thank you very much for the kind introduction and invitation by Sabine, who I actually know almost 20 years now since I was working at the Charité. And as you said, there I actually started with array CGH to establish this as a diagnostic method. In my talk, I will just present some basic principles and some examples of cytogenetics and CNVs in cardiovascular heart disease. I have no disclosures. And to start off, I want to point out that in the past decades, we have got more knowledge about genetic causes of CHD, and these are chromosomal aneuploidies, copy number variants, and recently also more single nucleotide variants. I will talk about the first two, and the next talk by Lorenzo Monserrat, I think he will focus on NGS data and the single nucleotide variants. How do we actually detect aneuploidies or copy number variants? These both require genome-wide analysis. For aneuploidies, we use the very traditional method, actually, conventional KU typing or chromosome analysis, which has a limited resolution, I have to say, but still it's beneficial to detect, for example, balanced structural variations, which can also be important. On the other hand, of course, the higher resolution that came up approximately 15 years ago is molecular karyotyping or chromosomal microarrays. You can use two different kinds. You can perform a microarray-based comparative genomic hybridization, where you actually compare patient DNA with a reference DNA, or you can use SNP arrays, which give you additional information, not only copy number, but also the SNP information. So that could be loss of heterozygosity. Recently, we have the possibility to perform massive parallel sequencing. So you all know whole exome or whole genome, and especially whole genome will catch up, but so far it's not in routine diagnostics. These methods, which are available, I point out here the possibilities and also the limitations. So you see from this table that none of these methods can actually detect all the aberrations. KU typing is focused mostly on aneuploidy and larger CNVs. The chromosomal microarrays are pretty good at aneuploidy and copy number variants, but both these methods do not detect single nucleotide variants or indels. This is actually what's been done by next generation sequencing. And as I already said, genome sequencing is catching up, but it's not in routine diagnostics currently. And I briefly mentioned FISH here. FISH is fluorescence in situ hybridization. This is actually a targeted analysis. So of course you can detect aneuploidy and copy number variation, but since it's not genome-wide, you only use it if you want to follow up specific questions, for example, in prenatal diagnosis. On this slide, I present chromosome aneuploidies, which means that you have an extra copy of a complete chromosome or a loss of a complete chromosome. And these are actually the most common aneuploidies, which are associated with cardiovascular defects. And I guess you are familiar with these syndromes. We have the trisomy 13, trisomy 18, 21, and also the monosomy X. These percentages actually relate to the frequency of cardiovascular defects at birth, I have to say. And most of these patients actually have a high mortality, especially the trisomy 13 and 18, and they will often die in utero, but still they show multiple birth defects. If they come to birth, then we also observe severe intellectual disability, but as I said, there is a high mortality. And Down syndrome, of course, is the one we know where there's also often cardiovascular defects observed, especially here approximately 50%. Monosomy X, there is a frequency of about 30% that we see heart defects in the live-born children. I point out recent reviews, so if you want to really go into detail, I'm not a clinician, about the cardiovascular features, then please look at these reviews. And I think we also have a nice presentation later on genetic syndromes in CHD by Answar Baban, so he will probably present more clinical details later. I want to show one actually rare trisomy, which is not on this common trisomy list. This is the trisomy of chromosome 22, which we observe frequently upon spontaneous miscarriages, rarely in live-born children. They usually die if they are born within the first days of life. Anyhow, I wanted to show this example because there is a high incidence of cardiovascular defects. Almost every child with a trisomy 22 has a CHD. And here you see a kayogram and nicely can figure out the 3 chromosomes 22. And there are some common extracardiac features also in the upcoming slides. I will also show these because these may be more interesting to the ones who see children, that you have extracardiac features and they often trigger the clinical diagnosis. So in these children, it's intrauterine growth retardation, for example, and microcephaly and other abnormalities. Coming to large chromosomal aberrations, which are still detectable by conventional karyotyping. The classical example is Jacobson syndrome, a terminal deletion on chromosome 11 on the long arm. I marked this here with a little red arrow, and if you compare the 2 homologs of chromosome 11, this can clearly be seen. So this is still possible to see and observe under a microscope. Frequency of cardiac defects is about 56%. And there are actually 2 critical genes. And one is the transcription factor ETS1, responsible for the cardiac phenotype. And a second gene, FLE1, which is responsible for megakaryocyte development and differentiation. And these 2 genes actually trigger the phenotype in this larger deletion. The other genes also contribute. And so we have a complex phenotype also with lots of extra cardiac features. And FLE1, for example, is responsible for the thrombocytopenia, which is observed in these children. Now coming to copy number variants, as I said, these are not detectable by conventional karyotyping because they are smaller than 5 to 10 megabases. So these are submicroscopic aberrations. They encompass deletions, losses, and duplications or gains. The pathomechanism behind copy number variants is usually the alteration of gene dosage, especially dosage sensitive genes. And we have another term for syndromes associated with copy number variants. We call those contiguous gene syndromes because the phenotype is usually made up by a combination of several genes that are located within the copy number variant. Another term you will find frequently in the literature is genomic disorders, and this relates to a mechanism which I will explain on the next slide. I put some numbers down here. You find different percentages in the literature, so I just took these 2 percentages. CNVs can explain cardiovascular defects in approximately 10% of isolated defects, and about 25% of the syndromic CHD are explained by copy number variants. Coming back to the term genomic disorders, what is meant by this? This actually means that these genomic disorders are due to so-called recurrent CNVs, and recurrent CNVs come back to a predisposing genomic architecture or so-called hotspots And these hotspots are actually consisting of segmental duplications or low copy repeats. And the aberration occurs because there is a misalignment during meiosis, and it comes to a illegitimate crossing over during meiosis. And as you can see in the scheme down here, this recombination then can lead to either a duplication of this region or a deletion. And these recurrent CNVs have identical breakpoints because of the log copy number variance, and they have identical sizes. So these are the genomic disorders. And on the next slide, I will show a few examples that are commonly associated with cardiovascular defects. The most prominent here, of course, is the 22q112 deletion. And again, the percentages relate to the frequency of cardiovascular defects. Additional genomic disorders associated with CHD, and they are below 50% of frequency. They are nicely also reviewed in those two recent papers. I don't go into detail of additional ones, so we can come up with approximately 40 genomic disorders, which also have CHD in their phenotypes, and I could not list them all. One important thing I want to point out already here is that we always have to consider incomplete penetrance for the cardiac phenotypes. So these are not always present, so this you should keep in mind. Now I show a few of the common ones and start off with the most common microdeletion syndrome, which is the 22q112 deletion, also known as DiGeorge or velocardiofacial syndrome. And this is the region which is affected. And you see here the low copy repeats marked where these recombinations occur. And the typical deletion is approximately 3 megabases. And this is the one where we also have the classical phenotype of DiGeorge syndrome. And the major gene is TBX1, an important cardiac transcription factor, which is included in this typical deletion, in addition to 39 other genes. You can also see that there are some atypical deletions located here in the more distal region. And this is important to know because these can also have cardiovascular defects. And if you do not perform a chromosomal microarray, but you only perform FISH, then you will miss these deletions. And I show the example here because the FISH probe that is used for detection of the deletion, is located here. And you can clearly see this only covers the typical deletion and the proximal deletions. Otherwise, the detection is nice, and you can use this, for example, in prenatal diagnosis. This is often requested if the ultrasound shows a cardiac defect, then we get this to follow up. And of course, FISH is a very quick and rather cheap analysis, but you could miss deletions. The second most common deletion syndrome associated with a cardiac defect is the 1p36 deletion. This is actually a subtelomeric or terminal deletion. You see the array profile here. You can see the deletion nicely here with the profile going down. And there are typical extracardiac features which usually allow a clinical diagnosis: microcephaly and growth retardation, and then some other structural brain abnormalities or hearing loss and skeletal abnormalities that often make it possible by a clinical geneticist to suspect this 1p36 syndrome, and then we can follow it up in the lab. I want to point out that cardiomyopathy, which we heard a bit about from Perry, is also observed in about 30% of patients with this microdeletion. And there is a candidate gene proposed for the cardiomyopathy, and maybe Sabine will talk about this later. Williams-Burdon syndrome is an interstitial microdeletion on chromosome 7 on the long arm, which is about 1.5 to 1.8 megabases. And the critical gene here is the elastin gene included in this microdeletion. And the elastin gene is responsible for the connective tissue defects, but also for some of the heart problems that we see in those patients. The other genes within the deletion are then responsible for the facial dysmorphism, for example, growth abnormalities, and especially endocrine abnormalities you can observe in these patients. What is very special about Williams-Burdon syndrome is the unique personality profile, and these people are really friendly. They are what we call party people because they chat, and so the intellectual disability can be compensated by this. Why did I put those two red arrows here in the profile? There you can actually see those low copy repeats that mediate this microdeletion, and these low copy repeats are flanking the aberration. So I just wanted to show this, and this is what we consider typical for genomic disorders. I'm running out of time, so I just briefly want to mention another phenomenon with those recurrent CNVs. You can have reciprocal CNVs, so you can have a microdeletion or a microduplication at the same locus, and they might share certain features, but they also differ. And one example for this is the 8p231, including the GATA4 gene, which is dosage sensitive. So there we can have both microdeletion and microduplications. And to notice here that the duplication has a milder phenotype and shows incomplete penetrance. To summarize, I hope I could show you that chromosomal abnormalities play an important role in CHD. We have recurrent CNVs, so the typical genomic disorders, but also non-recurrent CNVs with not identical breakpoints. And I showed you some examples of aneuploidies or genomic disorders associated with CHD. The molecular mechanism, especially for aneuploidies, remain largely unclear. We have so many genes so that it's really hard to figure out genes involved. This is a bit easier with the genomic disorders. There are some CHD genes identified, and often these are followed up and proven to be the relevant gene for the cardiac phenotype by using animal model experiments, for example, knockout mice, to show this. And in the end, which Perry also nicely mentioned, why do we want to have a genetic diagnosis? Because this genetic diagnosis clearly assists clinical management, and Perry pointed out some more reasons why you actually would like to have a genetic diagnosis for your patient. Thank you very much, and I'm happy to take questions.
Thank you very much for this really nice talk. Are there any questions from what I see? I think we can take one. So then I would go for one. So you mentioned mostly the chromosomal evaluation and and array-CGH. But what do you envision in the future for these patients? I think there are now techniques, and we'll come to this in the next talk, which are probably more useful in the overall setting. Can you elaborate a bit on this, how you do that and implement that in the clinical setting?
Yeah, I have to say what is actually implemented right now regarding NGS is in the routine diagnostics is exome sequencing. And I have to say, we see that about 30% can be explained by copy number variants. And these are really difficult to detect simply by exome sequencing, especially to set the right limitations of the CNV. You can detect a CNV by exome sequencing, but you can never precisely point out the exact size. So I would say, unless we really have genome sequencing in the routine diagnostics, you won't be able to figure out everything by exome sequencing. I think we need both. We should keep in mind that there is a contribution of copy number variants, and we need a method for this. And we have exome sequencing, of course, to figure out the single nucleotide variants. So I think we need still to combine both.
Thank you.